subject them to that and see what happens. So the mutants can be subjected to these drug-like compounds to see whether there is any rescue, or the wild-type embryos can be subjected to see what happens with when the embryos are subjected to these drugs. So there was no retinal model established for small molecule screen, so we wanted to establish that and look at two different processes, retinal vasculature and photoreceptor rescue. So I will not be discussing about photoreceptor rescue today because it is still going on. Retinal vasculature screen, we used transgenic FLE1 EGFP expressing fish and FLK1 as GFP expressing fish. So before I go into the vasculature, a quick view of how mammalian retinal vasculature develops. In mammals, early eyes is having two different vasculature. One is choroidal vasculature, another is hyaloidal vasculature. This is hyaloidal artery enters and the hyaloidal vasculature wraps around the lens. Choroidal vasculature is totally outside the retina. As the eye develops further, you can see that the hyaloidal vasculature regresses by a process called hyaloidal regression and is replaced by retinal vasculature. And the mature retina is characterized by choroid vasculature and retinal vasculature. Retinal vasculature again invades into the retina to form deeper plexus, inner deeper plexus and outer deeper plexus. So in zebrafish, it is not known. I mean, early retinal vasculature, development of early retinal vasculature is not known. So we decided to follow that. So in zebrafish, there are three different early retinal vasculature seen. One is intraocular vasculature, surface vasculature, and the choroidal vasculature. Intraocular vasculature is the first to develop, and you can see endothelial cells enter the retina via the choroid fissure, goes behind the lens, and sort of cups the lens and moves from the bottom of the lens to the top, as you can see here. So this would be the intraocular artery, and the lens would sit right into this cup. And by 144 hours, you can see that the blood vessels form linear arrays around the lens. And by 30 days, what happens is this linear array actually falls on top of the retina to form the mature vasculature. So surface vasculature also develops around the same time. You can see one single vessel opposite to the choroid fissure at 28 hours post-fertilization. By 72 hours, there are three different vessels which are connected into one circular vessel. We have named these three vessels as nasal, dorsal, and ventral ring vessel. And this basically stays the same at 9 days post-fertilization, and the intraocular vasculature actually connects to the ventral ring vessel as denoted by the asterisk here. Choroid vasculature, we didn't follow this type of vasculature, but we see that at 9 days post-fertilization it forms polygonal structures, totally enveloping the entire eye. So intraocular vasculature is very similar to that of hyaloidal vasculature, and instead of undergoing regression as mammalian hyaloidal regression, it undergoes remodeling. You can see there is a chaotic network of vessels at 48 and 96. By 120, you can see a sort of more organized form of blood vessels, and by 48, you can see a totally linear arrays of blood vessels around the lens. Blood flow is also detected by 55 hours. However, by 48 hours, you can totally see actually the network being established for blood flow. This is the retinal artery, intraocular artery, goes around the lens and then exits and connects to the ventral ring vessel. You can clearly see here how the blood flows. Blood comes in via the intraocular artery, goes around the lens and then exits from this vessel. So on the surface, blood comes in from the nasal ring vessel and then goes around and exits via the dorsal and the ventral ring vessel. So after repeated trial and errors, we found that looking at the intraocular vasculature at 72 hours behind the retina in this orientation would be the best way to assess would be the best readout for the small molecule screen. So we screened the library of around 2,500 compounds, and we found 5 compounds which showed retinal specific effects. The effects can be clearly seen when compared to the control. Enalateral malate produces thickening of vessels. Pyrogalin also produces thickening of vessels, but it also clumps the vessels. Albendazole and mebendazole actually reduces the number of vessels. The vessels collapses in mebendazole. even more than albendazole. Zearalenone also produces widening of vessels. So this is kind of retinal specific, as the trunk vasculature, which was taken as control, does not show any such effect. So we also looked at the retinal architecture by looking at different neurons and their arrangement in the retina. So you can see photoreceptors, retinal ganglion cells, and the optic nerve. So the green is the blood vessels. In the control, you can see that except for mebendazole, which shows extensive damage to the retina itself, all other molecules do not show any damage to the retinal neurons. So out of these 5 molecules, 3 of them were already FDA-approved drugs. Enalapril manganate was approved for high blood pressure, and albendazole and mebendazole are worm-specific drugs available in the market. So we wanted to see if we can actually quickly understand the structure-function relationship of these 3 molecules. And since enalapril manganate is quite well known, it is an angiotensin-converting enzyme inhibitor, or ACE inhibitor. So what we did was we designed morpholinos against ACE, and we injected it into the embryos. At 24 hours, you can clearly see that there is efficient knockdown of ACE. However, this knockdown is lost by 48 hours, where you can see the expression of ACE is restored almost in the embryos. So this inefficient knockdown can also be seen in the retinal vasculature at 52 and 72 hours post-fertilization. You can see that there is no real difference between the morphine and the control. So for the last 2 compounds, albendazole and mebendazole, they showed structural similarity. So we looked at, we scanned the databases to see if there is any more molecules which show similar structures. We found 5 with different sort of R groups, and these all the 5 showed similar phenotype, however with varying intensity when compared to albendazole and mebendazole. So you can totally see in the second half of the presentation that how retinal vasculature also develops rapidly, and in fact it begins even before the neurogenesis of the retina begins. You can also see that how extensive remodeling happens during the retinal vasculature development. So in zebrafish, there are actually 3 different vasculatures: intraocular, surface, and choroidal. As this vasculature also develops via angiogenesis, there was no hydroidal regression or deplexus in the fish, and we have also shown that this retinal vasculature can be used as a very good model for performing small molecule screen, and how easy it is, how rapid one can characterize these identified compounds. So I would like to sum up my presentation with a take-home message. Through this work, we show how quick the retinal vasculature develops, how efficient retina can be used as a model for small molecule screen, and how the identified compounds can be quickly characterized in the retina. We also show the characterization of 3 nuclear receptors, tailless, PNR, and LXR in the developing brain. We also characterize LXR and show its importance in the developing brain and the retina. In MEDARCA, we established the patterning of the retina and show that neurogenesis happens, 2 domains of neurogenesis starts in the retina. And we also show the timeline of retinal differentiation and compare it to that of zebrafish. So with this, I would like to acknowledge the contribution of certain people. From Soreton Hochschule, it was Lothar Hambrios, who has been very supportive. Karolinska Institute, Agneta Moth, my supervisor, who LXR work was done in her lab. Per Schulsten, Thomas Berglund, and Yurima Malitsky are my co-supervisors. TLX PNR work was done in Gieselbert's lab. Arindam is my mentor, and the rest of the work was done in Yurima's lab. I would also like to thank my friends and family members, funding sources, and